Thanks for having me here again at this series. I'm quite happy to be presenting again today and look forward to some discussion. If I could have the next slide, please. So what we're going to talk about today is the state of the possible in the current federated analysis environment. So this is essentially what's going on today in both in Canada and around the world in terms of federated data analysis. And we're going to talk primarily today about common data models. And I'm going to introduce the problem by talking about common analytic protocols versus common data models, differentiating between those two. And then talk a little broadly about the world of common data models and what's out there. And then I'm going to turn it over to Mike to talk about what CNODES has done over the past several years using one specific common data model that's been developed by the US Sentinel system sponsored by the FDA. If I could have the next slide. So we outlined, this is a bit of a review of the last webinar. We outlined in the last webinar a little bit of what we can do. In a federated environment, provinces, we could simply conduct studies in each of the sites and pool the results. That would be one way to conduct a federated analysis. We could, and I'm just getting a message on my screen that my internet connection is unstable. So if I do cut out, Alexander, please let me know and I'll work on this. We could conduct, and this is sort of CNODES' standard practice, common protocol-based analyses, where the protocol is designed at a central location in order to be as common as possible across all the sites. And the analyses are then conducted based on that common protocol and common statistical analysis plan. The next step up, and what we're going to talk about today, is what we'll call common data model-based analyses, where the data are standardized across the sites so that standardized analytic code can be run against the data. And I'll talk about a few different ways that can be done. And then finally, we referred to this a little bit in the last session, and we'll be talking more about these later on. Fully federated analyses like the data shield algorithms that allow communication back and forth between a data holder in one site and some central analyst that preserves privacy by not sending individual data, but sends data back and forth between the sites and the center, generating a fully federated analysis. If I could have the next slide. Okay, so these are the two that we're going to talk about today. This is just a schematic of the common protocol on the left, where analysts are running, the little Fisher-Price people in the bottom, analysts are running common analyses on each of the three sites and then sending summaries of those analyses back to the center, the coordinating center, who then summarizes those and produces a report. In the common data model frameworks, the work is done, there's a lot of work done at the site level, at the three sites in my diagram, to standardize the data. And again, we'll talk about different ways that we can do that, such that common analytic code can be sent out to the red site, the yellow site, and the green site, and run semi-automatically and then returned to the coordinating center for combining and then production of a report. Next slide, please. Okay, so let's talk a little bit about what this is, the common data model. It's a tool to standardize data into a common format. So it can start with structure, that is saying, let's have columns for ID, date, and other things, and let's format it in a very basic way. It could standardize format, so value sets in variable formats. Let's say, for example, we're going to standardize into ICD-10 codes, and we'll standardize more there such that you have the same coding structures and then the same formats in different sites. And then you could go further to standardize the meanings of the variables such that we end up with a common definition of, say, incident type 2 diabetes. And the common data model may also include standardized analytic code and analytics layers such that, as I was describing in the last slide, we can run things automatically against the different data sets. Next slide, please. So the common protocol approach that CNODES has run in the past has some advantages. Primary advantage is its analytical flexibility. You can start with a protocol that you then can design essentially any way you want, because you're just designing individual studies. You can design it to reflect different data sources within each site. You can design it with essentially whatever statistical methods you want, as complex as you want, because you're designing a single study. It also provides local capacity building because the analytic work has to be done at the sites by site experts. The limitations on this are obvious. It takes longer to write a protocol and send it around. And it risks inefficiency and errors because we're dependent on a lot more people in the process and a lot more quality checking, and there's a lot more variability in the process. Next slide, please. So let's look at why a common data model. The big advantage of the common data model is if the data sets across the provinces look identical and have the same structures, we can use standardized code. And when we have standardized code, we can make it reliable. We know it can be tested against the data formats. We know it's reproducible because we can just pull out the same chunk of code and rerun it against the same data set. We should get the same answer. It's rapid because we can pull from a set of available code pieces. It has guardrails. It tells us what analyses are allowed and what are not allowed. So it in some sense prevents inappropriate analyses. And at the end, you can generate standardized and automated reports. So you can actually produce or have the code produce a report saying, these were the methods run, here are the tables, without having to do much work on your end. Then when we're doing multi-database studies like CNODES and Sentinel in the US and Odyssey, we're running the same code across multiple sites. So we know that we've got this sort of site-wide reproducibility as well. Next slide, please. Okay, so I'm going to talk a little bit about different kinds of common data models. Mike is going to tell you a lot about organizing CDMs. And what we mean by an organizing CDM, and the Sentinel is the best example of this, is it reorganizes data files into a standard data structure. It doesn't modify the raw data content. So if it might say to, I'll just take a simple example of, let's say we have a database with procedures that were done. It will standardize everybody's data into a standard format so that it's ID, patient ID, date, provider ID, procedure. But it won't modify what's in the procedure cell of that data set. It will require you later on to, once you start a study, take that raw procedure codes and create the actual variables you want to work with, so project-specific coding. A mapping common data model, and the OMOP common data model is probably the best example of this, maps the raw data to concepts and constructs. So it will take it will first do that organizing step. Then it will take the second step of saying, we want to create a concept for incident type 2 diabetes. And the mapping can be study specific, but it's usually comprehensive and such that you bring your data into that OMOP format. And this we think of as a pre-configured rules system. And we're going to hear from at least one expert in OMOP later in the year. The advantage of this is that you've got really standardized data. The challenge is this mapping process can be an awful lot of work. Next slide, please. This is just a schematic of the kinds of CDMs that we can work with. So we have raw data along the bottom, and you may be in a relational database of some kind. It'll be some kind of large database structure. And then the next layer up is an organizing common data model, which just reorganizes the structure into a common format. And then there's a lot on this page that I'm not going to talk about, but then you can analyze these organized data in several different ways. You can do like OMOP does and create a pre-configured rule system that then you have this really heavily standardized data that goes to the analytics layer, and you know that you've got this sort of standardized coding of incident type 2 diabetes across different databases. You can have the adaptive rule system like Sentinel, where you then say, okay, I want to do a study. I'm going to create my variables by looking into that standardized data and saying, okay, with this set of ICD codes, I'm going to define incident type 2 diabetes, and then send it to an analytics layer where you can still use fairly standardized analytic code. Or you could take either the raw data set down at the bottom, or the organizing CDM, that is the structured data used by Sentinel, or the OMOP common data model standardized data, and write any code that you want to run the analyses. And so this sort of from left to right of this graph goes from the most automated to the least automated kind of analytics. And this paper referenced at the bottom is a very good reference on the kinds of CDMs that are out there, and you can read some more about some of the other models that are out there as well. Next slide, please. So I talked about a couple of common data models. We're going to be hearing next about Sentinel. There are some area-specific or subject-specific common data models, like the conception CDM for pregnancy and linked mother-baby data, because that in and of itself is a fairly complicated data structure and data setting. There are open research CDMs like PCORNet, which is a common data model that researchers can use, similar to the Sentinel, but with some differences. There are commercial ones where individual companies have created their own common data models and are working with their own software. And then there's OMOP and Odyssey, which is a large group of researchers. We talked a little bit about this before. And the OMOP common data model has this standardized mapping approach. This is the approach that's being taken by the Darwin EU project, which is the European equivalent of CNODES, and it's also taken by the Odyssey group, which is an open source community of researchers, both academic and industry researchers working together to develop these data. Next slide, please. So common data models come with a set of analytic packages. Sentinel and OMOP are open source SAS and R code respectively. Almost all of the code from both are available open source on their websites. And then there are proprietary platforms like Edion and others that are working behind their sort of commercial firewalls on their own code. The analytics, the main benefit of the analytics, and it's a big one, is that it increases speed, reliability, and reproducibility, as we discussed. On the other hand, it does somewhat limit flexibility. I did describe how you could use the common data model with any code that you want, but the basics are what's pre-written. Next slide, please. So I'll skip over this slide very quickly. You've heard about CNODES a lot. We use population-based administrative healthcare data to provide responses to queries from stakeholders regarding drug safety and effectiveness. Next slide. CNODES as a whole, again, involves data from across the country and from the UK. We have a database development team led by Lisa Licks, whose role is to work with organizations like HDRN and others to work on problems like we're talking about today. Next slide, please. Here's our typical data, which are the standard provincial data sources. And since CPRD is an EMR-based data set, we also have some more clinical variables measured there as well. And with that, I will turn it over to Mike, who's going to talk about the next slide about the Sentinel Common Data Model specifically and how we've been using it.
Thanks very much, Robert. Alex, if you wouldn't mind just turning back just for a second. I'm just going to recap the core administrative data sources that we're going to talk about that we've used for the development of our Canadian transformation of the Sentinel Common Data Model. So on the left-hand column here, we have our core provincial health administrative data sources, the provincial health insurance registries, prescription drug claims, physician service claims, hospital discharge abstracts, emergency department records captured in Canada largely in the National Ambulatory Care Reporting System. We also have same-day surgery records and vital statistics records. And these are the primary data sources that we'll be talking about today that we've used to develop our common data model tables. On the right-hand side here are a number of supplementary data sources that have been used in our protocol-based analyses, depending on the nature of the question being asked. For example, our cancer registries for cancer outcomes. And as Robert said, the clinical practice research data set, which is a UK EMR-based data set that has allowed us from time to time to bring in other risk factors as potential covariates and for confounding control that we wouldn't normally have access to in our administrative data. But these on the right-hand side are not are typically used in our common data model projects. Next slide, please. So when we talk about the FDA Sentinel Common Data Model, we're really referring to two main components. The first is the set of standardized, regularly refreshed common data tables. I'll walk through those tables in a second and the underlying data sources that we've used to construct them. And then secondly, as Robert said, a set of reusable, flexible modular SAS programs. And I'll talk about some examples of these in a minute. But in essence, they're a suite of programs that are customizable to answer a set of commonly asked, in the case of the Sentinel program, drug safety, drug use questions. So these span from looking at numbers of newly exposed individuals or specific periods of time nested within specific diagnoses, for example, clinical cohorts, crude outcome event rates following new drug exposure, and then a variety of drug use programs looking at concomitant drug use, persistent switching, et cetera, and more sophisticated designs that I'll talk about in a minute. Next slide, please. So this provides a high-level overview of the Sentinel distributed data set and the common data tables within the entire sort of globe of data that the Sentinel team have developed. For our CNODES projects, we focused primarily in the upper left-hand corner here, the administrative data tables. The enrollment and demographic tables draw primarily from the provincial insurance registries. The dispensing table draws from the prescription drug claims. And then the encounter, diagnosis, and procedure tables draw from multiple sources. So all of those encounter-based sources I talked about earlier, the physician service claims, the discharge abstracts, same-day surgery records, emergency department records, and all of these tables are linked at the patient level using a unique patient identifier. In Canada, typically a coded version of our health insurance number. And then for the encounter, diagnosis, and procedure tables, these are all linked through a unique encounter ID. So they're linked across the different contributing sources here, the diagnosis codes, procedure codes, and so on at the encounter level. And then in the lower right-hand corner, lower left-hand corner, we have the death register, which is obviously critical to incorporate death in as an outcome and censoring factor in our analyses. And then another that we haven't yet used, but we've developed in multiple centers, is the mother-infant linked data set. And this is essentially a linkage of mother and baby records from live births and derived from the hospital discharge abstracts. So that's one that we hope to use in the future. Next slide, please. So this is a schematic that provides a nice overview of the process that the FDA Sentinel team use in carrying out query analysis. So the first step really is to take those modular programs and populate them with the codes that are used to develop the cohort, the covariates, the drug exposures of interest, the outcomes of interest, and then test that program on a local data set. Typically, they would use the US market scan data set in a standalone version, transformed into the common data model. Once that's run successfully and the results are as expected, the package is then distributed to all of the data partners, and the Sentinel system involves 18 or 19 different data partners, and they're all primed and ready to receive the package and run it against the local versions of the common data tables in step 3. Once the results of the query, these are standardized, as Robert said, standardized outputs that we would expect based on the initial run. Once those are received and reviewed, they're then securely transferred back to the coordinating center in step 5 through a secure network, where the operations center then pools the results across the various centers using a separate aggregation SAS program that we've also used within CNODES. So really, this is the process that we've attempted to emulate within CNODES, obviously working very closely with the Sentinel team. All of the resources are in the public domain, and I'll talk about some of those in a second. Next slide. So all of the projects query projects that are undertaken within the Sentinel system, as I say, are in the public domain. A couple of slides here. I'm just citing a few specific project examples. You'll be able to go to those sites, see the reports, and for more recent projects, the actual program code, pre-populated program code that could then be modified and run locally. And it's these packages, so to speak, that we've used in the replication of Sentinel projects here in Canada. So the first one is really just looking at concomitant drug use. The second, characterizing a cohort of patients and describing patterns of drug use in that cohort. The third one that we've run multiple times in CNODES is simple drug use cohort and crude outcome event rates following new use. Next slide. And then they also have a suite of active comparator analytic packages. So these would include propensity score matched cohort studies, propensity score weighted analyses. And here's simply an example of a comparative analysis of oral anticoagulants and GI bleeds that leverage the propensity score matched cohort design. And then also a series of self-controlled case-only analyses. So those are also available. So as I say, available on the website if you'd like to have a look at the reports and then corresponding packages in a GitHub site that they've developed there. Next slide, please. So one of the things that came up in the process of the interim evaluation of our initial CNODES grant was the recommendation that we look for opportunities to improve efficiency. And as Robert said, one of the key advantages of a common data model is timeliness, efficiency, reduced risk for error. So as a result, we sought additional funding to develop the common data model tables across the CNODES sites that Robert described, initially in phase 1 in Saskatchewan, Manitoba, Ontario, and Nova Scotia. And in the course of this project, we spent several months developing the common data tables at each site. Sentinel provided us with QA packages that allowed us to run SAS code against the tables to make sure the contents was accurate, that outliers were dealt with, and so on. And then when the tables were constructed after a few months of groundwork, we were in a position to run a series of pilot projects, which we did. Again, replications of projects that had already been run in the Sentinel system that we'd adapted for Canadian questions and drugs and DINs and so on. So for example, the first one was a simple drug use analysis of new molecular entities approved by Health Canada in 2015 and the uptake of those agents over time in the study provinces and so on and so on. Next slide, please. So this slide provides sort of an overview of the results in terms of timeliness of phase 1 of our demonstration project. So column 1 reports the review times for the data custodial reviews and REB reviews at each of the sites. As you can see here in Nova Scotia, we were able to negotiate a commitment to a 5-day turnaround time for these analyses. And it ranged from there up to about 50 days on average in Manitoba. So quite a range of approval times. And this continues to be a challenge for us. The Ontario site served as the essential pre-pilot site in the sense that we populated the SAS programs with the input files that we needed and ran them, tested them in Ontario, and then distributed those to our 4 sites or 3 other sites. And then over the course of 2 weeks or so, those were run. They couldn't be shared with us for aggregation, however, until we sorted out a protocol, essentially a policy for how we would share these small cell tables, because at the end of the day, these are small cell summary tables or SAS code output files that have to be aggregated. So typically it would not be permissible to share that sort of data across provincial lines. And so we had to negotiate protocols for that. We were able to do so in Nova Scotia and Ontario using pre-existing policies in place for sharing of small cells. In Saskatchewan, they also had a process in place for doing so that involved the data custodians reviewing these sample outputs on a case-by-case basis and approving them for release. In Manitoba, however, it was not permissible to share. And so as a result, the Manitoba site, the Manitoba Center for Health Policy, became our de facto pooling site. So we could share our small cell tables with the Manitoba Center, and they were able to run the aggregation programs on behalf of all of the sites. And typically that would just take a few days to achieve an average turnaround time in the range of 80 or 90 days for these pilot projects, which was certainly an enormous improvement over the protocol-based timelines that we were seeing prior to that. Next slide, please. So there are a number of lessons from our initial pilot project. Obviously, the data custodial and RAB approval times are long in the grand scheme, and continue to be a barrier to more rapid, timely analyses. We obviously have to continue to negotiate shorter approval times. We've been successful in some cases and not so much in others. We did reach out to INES, the technology assessment team in Quebec, to explore the possibility of developing common data tables for Quebec. We continue to discuss the possibility of that, and it's a possibility, but we haven't successfully done that to date. And then also there are a number of tables that are missing from our suite of tables that we had hoped to develop. We did add one for COVID PCR tests for our COVID studies, but there are others that still are yet to be developed, and we'll look at that on a case-by-case basis down the road. Next slide, please. So in following from our pilot projects, we launched into a series of international collaborations with the support of Health Canada and the regulators in Europe and FDA. The first one was a study that looked at angiotensin receptor blockers and the impact that the nitrosamine recalls had on utilization across the countries. The later two are COVID related and grew out of an international consensus of the regulatory authorities. A work plan was developed, a COVID-related work plan was developed, and a series of studies grew out of that work plan, the two that we undertook here across Canada, the US, and Europe. One focused on the use of corticosteroids in mild cases, outpatients with COVID. That was undertaken in Canada and the US. And then the final study that we've just wrapped up is a study of coagulopathy in patients with COVID, both in the inpatient setting and the outpatient setting, and that was across the 3 continents. Next slide. So this just gives you a flavor of a piece of the results from that first study, the angiotensin receptor blocker recall project. And this plots the monthly proportion of new ARB users over time throughout the period of the recalls, which were summer 2018. And you can see here the impact that the recalls had on the main ARB that was affected, valsartan, particularly in the US and Canada, dipping down in the summer of 2018 and not recovering after the recalls. Very little impact in the European countries, primarily because of the mix of ARBs that were used, the market share of ARBs in Europe being quite different than we see in the US and Canada. Next slide, please. The second study was a study that grew out of the UK-based recovery trial, which some of you may be familiar with. And it showed that hospitalized patients who received dexamethasone in hospital had improved mortality rates, Subsequent observational studies in less severe cases of COVID in the outpatient setting were not able to replicate those results. And so recommendations generally were to not prescribe steroids in outpatients in milder cases. And so the regulators were quite interested to see whether or not that advice was being taken up in the outpatient setting. Next slide, please. So we actually undertook independent studies in the US and Canada, leveraging the US Sentinel package. The left-hand panel here on the left of the left-hand panel showing the uptake of steroids, the prescribing of steroids within 14 days following infection among outpatients, the left panel being Medicare patients, seniors, the right-hand panel being all of the Sentinel, all the other Sentinel patients collectively, and showing that by, say, August of 2021, roughly 20% of Medicare patients and about 14% of all patients were receiving a prescription for steroids within 14 days of infection. We contrast that with the right-hand panel in Canada, where by early 2021 in Ontario, which is largely seniors, we reached a peak of about 9% of patients receiving a steroid prescription within 14 days. And contrast that with the other two provinces that were involved, BC and Manitoba, where the rates were around 2% at peak. Next slide, please. The last study that I mentioned is a study of the natural history of thrombotic events in patients with COVID, both in the hospital setting and the outpatient setting. We looked at both arterial and venous thrombotic events within 90 days of infection. Next slide, please. And these results will be coming out very soon, just accepted. And they show quite a bit of heterogeneity across the countries. In the upper left-hand corner, blue, showing the 90-day risk, absolute risk of venous thrombosis, red arterial thrombosis over time during the course of the pandemic, pre and post-vaccine eras. In Canada, upper left there, the rates were among the lowest in the countries that we studied. On the lower right, the US showing considerably higher rates. And again, this is in a setting where we use the common data model, the Sentinel common data model in the US and Canada. And then the European countries use the OMOP common data model that Robert referred to earlier. Lots of reasons why we might be seeing this sort of heterogeneity. One important one comparing Canada and the US is the inputs to the definition, the case definition for COVID. And in Canada, we used exclusively PCR lab tests to define our COVID cohorts, whereas in the US, they used outpatient diagnostic codes together with lab data, which unfortunately weren't comprehensive. So they had to cobble together a broader definition. And that, among other factors, would have contributed to some of the variation that we're seeing here. In the European data sets, another factor would be that some of these are EMR based, whereas in Canada and the US, they're exclusively administrative data based common data models. Next slide. One of the nice things about collaborating with our US colleagues is the complementarity of the data in the two systems. And so while obviously the population size is multifold larger in the US, we have advantages in Canada as well. So you can see here on the left-hand panel, the distribution of an enrollment time in the US Sentinel distributed database. Median enrollment time there with churn and change among insurance systems is 4 to 5 years median. Whereas you can see on the right-hand side, just a snapshot of the follow-up time in Manitoba, where almost 50% of the population has over 20 years of follow-up time. Obviously, there are advantages to that sort of follow-up time, and I think we can work together with FDA to help support questions where that sort of follow-up time, cancer outcomes, for example, might be advantageous for us to be involved in studies. Next slide, please. So following on from those projects, we have a couple of new demonstration projects underway now. And these are exposing us to new analytic packages that we've not used before. The first is a drug safety signal detection software package called TreeScan. And we're using that particular package in a case study, a matched cohort study of patients with diabetes receiving the anti-diabetes weight loss drug Ozempic in family. And you probably will have heard of that class of medications, GLP-1 inhibitors. And then the second one, there was is a new series of projects looking at drug use during pregnancy. The first case study, anti-diabetic drugs during pregnancy, which is a new area for us, and we're looking forward to develop skills with that mother-baby linked data set. Next slide. So just to wrap up, a few thoughts looking forward for CNODES and the Common Data Model. Obviously, we have work to do with our project approval times. We continue to work on reducing those. Unfortunately, in some sites, they've actually gotten longer rather than shorter as a result of COVID and staffing issues at the sites. That's an ongoing challenge for us. It comprises a good chunk of the project time, and we want to try to reduce those to the shortest period possible. We continue to explore opportunities to acquire the Quebec data for common data models studies. And then finally, we want to expand our experience with some of these more novel data sources and packages, like the TreeScan software that we're using in this Ozempic project. So that takes us to the end of our formal presentation, and we'd just like to acknowledge our funder CoLab and Health Canada, and obviously the partners that we work with at the Sentinel Coordinating Center that help support our work with the Common Data Model. Happy to take questions.
Thank you so much, Michael and Dr. Platt, for that great presentation. Yeah, we're now moving to the Q&A portion of the presentation. So please, if you have any questions, post them in the chat and get started. So I think the first question is from Georgina. Could codes be converted from one format to another, for instance, from CDM 9 to CDM 10, or from ENCODE to CDM 10, and vice versa?
Do you want me to jump in on that one, Robert, first?
Sure, thanks.
So we've actually, in the course of developing the initial version of the tables that we're using, version 7, went through the process of developing some code conversions even in that version, let alone across versions. So, for example, our physician service claims are all province specific to the schedules of benefits in each of the provinces. And it was important for us to develop a common platform, a standardized approach to those across provinces. So we worked closely with CIHI. They had developed a crosswalk for their schedules of benefits across the provinces and territories, and we were able to leverage that. So that was one place that Robert talked about where we did take advantage of that crosswalk to standardize across provinces to a uniform coding system. And then as far as our movement from version 7, which we're currently operating in, to version 8, there are a number of changes and new tables and data elements that we will have to move to. One of the challenges with that translation is moving away from our health insurance, our coded health insurance number, which is site-specific, to a uniform approach to the unique patient identifier that's agnostic to the local provincial health insurance numbers. So that's something that we will have to tackle over the next year or so, is moving to that version 8. Most of the packages that Sentinel has developed to date have allowed for down versioning. So we've been able to operate version 7 and run those packages from this point forward in 2024 onward. They will not be back compatible, so we will have to move to version 8.
Thank you very much. The next question is from Michael. For admin data sources, the admin data sources you were listing, to what extent are they linked at the, I think that's provincial territorial level?
So at the
I can-
provincial, sorry, go ahead, Robert.
I could jump in and answer this, and then because it looks like it's, Michael, you've got 3 questions that are kind of linked together here. They are at the provincial level, they are linked data, as you said later on. They are linked data. And where's the privacy confidentiality assurance step? And then the third question was about small cells and disclosure. Essentially, what we've done within CNODES is follow whatever the provincial sites, since it's the provincial sites who are conducting the analyses and releasing the data, we've followed their guidelines for small cells, the standard guidelines at ICES and at Manitoba Center, for example, in order to make sure, and it's at the responsibility essentially of the site to follow their own procedures to make sure we don't send small cells or send potentially disclosable data to the coordinating center. Further to that, what CNODES has done across the board is our goal is to say whatever the minimum, or whatever, sorry, the maximum most restrictive small cells policy is across all the sites, we'll follow that across all of our sites. So we'll push it to a next level and say don't disclose under whoever has the most restrictive policy. Mike, I don't know if you want to add anything to that, either from the ICS perspective or from yours.
No, in fact, yeah, we for our protocol-based analyses, obviously we're using the lowest common denominator for sharing small cell tables that are suppressed and cleared at the local level and then shared with the coordinating center. Again, as I mentioned earlier, we have developed special protocols for the sharing of small cell tables. So these aren't suppressed tables for the purposes of aggregation using the common data model. And so those are very specific to the common data model output files and the pooling of those output files, and distinct from the protocol-based analyses. So they're special and follow the protocols that we've agreed to at each of the sites.
Thank you. We have another question from Amani. Was the ethics approval for creating the CDM linked to a specific research question? And if so, how do you maintain CDM for use in future research projects?
Would you like me to jump in on that
Yeah,
one?
I was going to say, do you want to take that one? Because you have more.
Sure. So at all of the sites, we initially launched our pilot project with a general ethics approval and protocol that allowed us to construct the tables at the outset and run a series of demonstration projects. So that was, in essence, the protocol for the pilot project. And then subsequently to that, each individual use of the common data tables has its own separate analytic plan and protocol that must be approved at each of the sites. So For the purposes of the timelines that I talked about separately, each of the projects within the initial pilot were reviewed independently, just as we do now post-pilot. But to answer the initial question, there was an underlying infrastructure submission and then subsequently individual project submissions for each query.
Thank you. I think we have time for one last question from Georgina. Is the conversion document that you were speaking about public and available, at least in CaiHi?
So feel free to write me and I can link you with the folks at CaiHi. I'm pretty sure that it's freely accessible, but I can certainly connect you with the folks at CaiHi who shared that with us. And hopefully it's useful to you.